Morning to everyone. Uh, Thank you so much to Dr. Anandan for very kindful introduction. And I'm very happy to be here. And it's my great pleasure to present my some of the recent works in front of you. Today's talk, I'm focusing on some of remarkable interactions between insects and bacteria. In this talk, I cover just two slides of introduction. Then I will talk about three different works which are common in the sense of showing some influence on the adaptive radiation or adaptive evolution of insects. And finally, I will briefly tell about what I'm doing in Harvard right now. Okay, and the most of the works I'm showing today done with Professor Boswell and Professor Edward Jacques from Hebrew University of Jerusalem, Israel, and my current group leader, Professor Cassandra Extover. So we all know now the literature from literature we are all know a lot of uh, studies coming out about the microbiomes, right? The microbial threads are really woven into the fabric of multicellular life because microbes everywhere. Often I say like we are living in the world of microbes and they are not in our world. So we are living in their world because they made this world through they help a lot for the evolution of Earth. So yeah, so I try to say the microbes are very important and they are real heroes of the Earth. So we all carry a lot of microbes, right? Every human or every multicellular organism carry a lot of microbes. And this one key thing we can do, you can understand the importance of microbes in the evolution. Our mitochondria, the powerhouse, is actually evolved from the microbes. Now the study initially it was that theory. Now many many evidence coming up to prove that. And if you see the microbiome, like we can differentiate the classify the microbiome in two categories when we talk about microbiome of insects. One is like primary symbionts or primary microbiome, and the secondary symbionts or secondary microbiome. This primary microbiome we call like obligate. They are without those microbes, either that insect cannot live, or without that insect, this bacteria cannot live alone. So they usually they provide like nutrients otherwise unavailable. The secondary symbionts are secondary microbiome, usually facultative symbionts. They are living mostly in the gut or surface of any in host body, and they are also providing essential nutrients and additionally they provide a lot of additional beneficial traits for like improving the reproduction fitness and yeah, and the reproduction is the key for the evolution. So so secondary symbionts also very important. So with this, my interest, my studies recently from last 6 years, I'm focusing on different aspects of host microbe associations, all relevant to adaptation or some way evolution. So the first study is about adaptive expansion and contraction of microbiome in polyphagous insect, which is Ceratidis captata. This like it's a very deadly pest, but it's a very good study system also. This fly belong to this Tephrichidae family, and within this Tephrichids, we can see different kinds of flies in terms of their specialization for the host, like host range. For instance, this polyphagous fly, the Ceratidis capillata, can oviposit more than 350 host plant species. But oligophagous is flies can develop or oviposit only plants within one particular family. The one example is that Darcus ciliatus. So always prefer only Cucurbitaceae family. And the last one, the monophagous fly, they are specialized to one particular fruit, particular species, plant species. So one example is this Bactrocera oleae, and its host species, oleae species. The interesting thing is within the family, these different species with a different range of host range, with a different host range. So my interest is how microbiome can favor this kind of monophagy or polyphagy. And if you look at a little about this monophagous fly, for this Bactrocera oleae, commonly we call it as olive flies. They harbor this endosymbiont. It's an obligate symbiont called the Erwinia duscola. It's a belongs to Enterobacteriaceae family. This bacteria provides nutrients and detoxify the phytochemicals which are very toxic to the flies, and also it's very important for the fitness. In one of my study, I confirmed that without these microbes, even these flies cannot identify the fruit perfectly. Actually, when I present different kinds of fruits with different colors, with the same size, without microbiome, with microbiome, and with the defined microbiomes, I found that specifically they prefer green olives, but if you disturb its microbiome, then they are losing its ability to directly target olives. So it suggests the importance of microbiome beyond this other detoxification or nutrient provisioning. It's beyond that. It's up to even like connecting the brain and gut. So it's like up to the even to the behavior level. And there are many studies relevant to this in the human system. Like if you transplant the microbiome of autism guy to normal guy, you will see the some kind of autism behavior in the normal person. So that brain gut axis already established. And the on the other end, this polyphagus fly, Ceratidis, again they harbor Enterobacteriaceae without any endosymbionts, but these bacteria providing nutrients, and it's very important for the reproductive fitness. If you remove the bacteria from this fly, then within 2 generations they will like they can't survive beyond 2 generations. If we look the how the microbiome transferred from the mother to offspring, it has a very defined pathway. When this mother fly oviposit, it smears the gut microbiome on the head. The hatching larva immediately consume those bacteria, so those bacteria establishing in the gut. So that's the passage pathway because different insect has different uh, mother to offspring transfer pathways. If we look at its life cycle, we can see two different life stages, very different life stage, active life stage. One is adult stage and larval stage. In the larval stage, they are living inside the fruit, but in the adult stage, they are free to choose their food. If we look this, like they evolved to adapt in these two conditions, right? In the one is this one condition is introduced by the developmental difference. The second difference is by the larval host. Because mother, each time the mother chooses the fruit. Maybe in one generation, maybe they are choosing one fruit. The next generation, maybe if the fruit is not available, then it will go to another fruit. So these two kind of effects can affect the microbiome. So so I wanted to check how this microbiome dynamically works to favor the polyphagy in the different fruits. Yeah, for that I set up a multi-generational field-like experiment. That initially I collected the female flies from the field collected fruits. Then in the first generation, I introduced those females to particular type of fruits. Then I collected the offerings from the that generation. Then again, I transferred to the different fruits in the second generation and so on, up to the third generation. Through the 6 amplicon metagenome study, I asked many questions about this. Why microbiome changing? Who is the controlling factor to this microbiome change? So firstly, when I look the microbiome diversity, if we, in this plot, you can see in the x-axis the Shannon index and in the y-axis kernel density of that Shannon index. You can see the difference in the normal stage in the F1 normal in the mother fly F0 mother fly. The microbiome Shannon index is within 1 to 3 feet. But if when it was their offsprings, their direct offsprings had microbiome, it is the diversity from 2 to 7, 2 to 6.5. So there is a very significant expansion of microbiome from mother fly to larval stage. If you trace that further to the next stage, that is like young adult stage, like tenorals, I collected like within 24 hours of eclosion. So the stage next to the larva, right? The larva then pupating, then from pupa the young adults coming. In the young adults, the microbiome again returning, it's like contracting. It's you can see the second because the non index of larva is this one like 2 to 6.5, but general microbiome Shannon index it's 1 to 6. It's slightly reduced. If I look again from the terminal to the next generation mother, again the microbiome shrinking and it's going back to the original. The mother fly microbiome was 1 to 3, now again the 1 to 3. So this pattern I noticed in all the generations. So it's a clearly says there is a strict microbiome expansion and the contraction across the generations. So then I wanted to check who is the controlling factor, what are the factors affecting this. So for that I selected only the F0 mother as well as the F1 offsprings and I checked the microbiome. In this plot I show the in the x-axis we have NMDS plot, NMDS. It's a NMDS plot. You have NMDS 1 and 2 in the x and y axis. In this, the clustering usually defines the difference. For example, if you see here, I have the groups. Like each group represents a different fruit, and you can see the differences. Actually, this looks a little complex, but when I look through the statistics, the difference between the host is very significant. And so, yeah, it is on the same generation. So if you look this again in the like if a same fruit, if you provide the same fruit in the subsequent generations, two successive generations, the microbiome is same. So it means the micro if the fly develops in two generation in on the same fruit, the same microbiome is there. But if in the different fruits, even in the single generation from same mother, they have different microbiome. So it means the effect of fruit as well as the mother. I will show that in detail. Then I looked the in the particular fruit. For example, in the particular fruit, apricot, the microbiome of offsprings from three different flies showed different microbiome. So it is like these different offerings from three different mothers. So it's a effect of maternal effect, maternal microbiome. So that even we can see that that effect in all the fruits. And in some like in loquats, it was not such case. One fly, like in one mother fly had different microbiome, their offspring had different microbiome, but others clustered little showed little similarity. Even in the Pitango, uh, some had like the two offsprings from two set of offsprings from two different mothers have mothers shared the microbiome, but this offsprings from another mother had different microbiome. This all summarizes that there are two factors controlling the microbiome here, the maternal and host fruit effect. If you look the effect of mother fly alone, the effect is the influence is only 7.3%. So that is contributing for the variation in the system. And the fruit represents 12.6, but the interaction between these two factors is 51%. So it means this it's the uh, dynamic interaction between the fruit. So both together defines the microbiome of the offsprings. So again, in this study, I try to trace the indicator species or some like the from where the microbe coming and where disappearing. And if we look the motherfly microbiome, it had the Klebsiella as a dominant species and followed by some unclassified Enterobacteriaceae and again another Klebsiella species. When we look the larval microbiome, the additional species like Gluconobacter started appearing, and in the terminal stage, even this Gluconobacter and other species expanded more. But again, in the subsequent in the subsequent state or F1 mother state, again the microbiome is similar to the previous generation mother. So Klebsiella and the Enterobacteria and another Klebsiella species. Again, in the second generation, the larval microbiome had this Gluconobacter because this Gluconobacter in some way introduced from previous generation food. Now it's established here, and it favored by this fruit again. So so these all different analysis showing that the two factors strictly controlling the microbiome. So that this difference because this fly should face this different fruit chemistry. So that is favored by this bacteria. And additionally, we did some metabolic potent predictions. So this actually called quickest analysis. In this, you can use the sixness data as a proxy. If you're using the sixness, then you can extract or uh, collect the close related genomes. Then you can look those genomes for the functional importance or metabolic profiles pathways, so that we can predict the function of those metabolites, that microbes in the system. So in this plot, again, it's an NMDS plot and based on Breakattis matrix. And here it shows different metabolic profile of the microbiomes living in these different life stages. So if you closely look the pathways, I found some notable pathway abundance in some cases. For example, in the low part, I found the pathways of vanillin and vanillin degradation, and this because actually this low cut in the low cut after harvesting, lignin usually establishing like lignin quantity become very high in those fruits. So I used I did experiment in the lab. So I used harvested fruit. So so my explanation for that is like so because of vanillin, this lignin abundance in the fruit, these pathways enhanced. And again, these different like in other fruits, we found the high expression of monoquinone or like other vitamin by synthesis pathway. So, so it's a depends on the fruit, the microbiome enrich, and depends on the microbiome, they are helping to the host. Here the conclusion is mother fly goes to the fruit, and while inoculating the egg, it introduces again it's a close to the mother fly. It's like say the microbiome is changing. Yeah. So the second study is about so now we look the outside of the fly and fruit. Now, if you look, little zoom into the fruit, what is happening inside the fruit? Here, inside the fruit, as I said earlier, the microbiome expanding, and these larvae having the feeding activity inside. And what is going on between this? Is there any interaction between the bacteria or this larva? And if you look there, you can see we can see some kind of cooperation by because of the feeding activity of larva, the surface area for the microbial colonization increases. So it smashes the fruit inside, so going wandering around the fruit, inside the fruit, so that it increase the surface area for the bacterial colonization, and the bacteria metabolizing these fruit chemicals and making it mobilizing it for the insects. So we expected some kind of attraction or interaction between these fruits, this microbe and the uh, larva inside the fruit. So to test that hypothesis, we had a hypothesis like the larva orients towards certain bacteria, and this response can be modulated by the symbiotic state of larva and by the microbial exposure. I can explain in the detail in the figures. Here we set up an experiment in that we use the medfly lab colony, and from that we collected the eggs and we sterilized some eggs as to generate oxalic eggs, like uh, the bacteria without bacteria. Sorry, the larva to get the larva without bacteria. And another is like symbiotic larva. It means the larva with to get the larva with a native bacteria. So when we put these two kinds of larva on the petri dish with a bacterial colony with 1 centimeter distance, you can see different pattern of attraction. Like if you actually this system works like if the larva touches the bacteria, then it will move around. So next day we can see the trail, right? The bacteria touching the larva touching the bacteria, then moving around. So the bacteria will grow on the media. We use the nutrient dot for this. So they are moving around. So we can see the trail, whether the larva touched the bacteria and how much it traveled in that way within fixed time. We use like 16 hours incubation for this. And we found that the different bacteria show different effect. Like for example, the pectinobacteria, while in the symbiotic ones, it's a similar in both cases, but they showed preference. In the Pectobacterium species, when you put the Pectobacterium species, the larva goes both arsenic and non-arsenic going like to both sides. Like again for the Klebsiella, in the symbiotic ones, we found like very high attraction towards the Klebsiella. But when you remove the that microbe, remove the microbes in the larva, they are not going to the Klebsiella. So this, yeah, in this particular case, it means the if some bacteria already there in the larva, then their preference is changing. Right? So if it is with some the bacteria coming from that, then they from the mother, that symbiotic bacterium, then they are preferring the Klebsiella. So, yeah. And the pneumonia, Klebsiella pneumonia, it's another species, but it's the pattern is same for both larvae. But this is the striking result is this against the Pseudomonas and the Staphylococcus. They showed very little preference to these two bacteria when compared to all other species. The key thing is, except these two, these all bacteria commonly found in the medfly. But these two are rarely we are isolating from the medfly. By evolution, maybe these bacteria, their preferred bacteria already they are established for cooperation, but here no. And we wanted to confirm further how is it real? The attraction is real. If we for test, we real with a pre-exposure or different conditions, we use like a two-choice experiment. Here we provided like two choices in the second level, and the larva in the center with the 2 centimeter distance from the colonies. And after the incubation period, we found that if you if you put the larva already treated with the pectobacterium, yeah, if you already treated with the pectobacterium, they are preferring pectobacterium, and it's equally going to the Klebsiella also. If you treat, if you have the larva treated with the pectobacterium, and then if you have choice of pectobacterium as well as pseudomonas, then they are preferring only pectobacterium in all cases except the oxenic conditions. So they are not preferring the pseudomonas if they are symbiotic or treated with some specific bacteria. And in the last case, again, if you provide the staphylococcus with the pectobacterium, all strictly going to the pectobacterium and always creating this staphylococcus. So this is another pair. We use the one preferred for Providentia and other side different bacteria. So again, the pattern almost the same, like Pectobacterium. They are avoiding the Pseudomonas and the Staphylococcus, and that avoidance also depends on the condition. If they are oxenic, they are not showing any such difference, but if they are treated with some bacteria or symbiotic, they show different habit. So, and another thing here to confirm whether really the bacteria going towards the it's a because of bacteria or something else. So for that, like we measured the trail length, just like with bacteria or without bacteria. When we put the bacteria, their movement increasing. It means their activity is enhanced in the plates with the bacteria. So it confirms that the movement is because of some bacteria that we are putting in the plates. And then we wanted to check the mechanism. What is the mechanism? What's the mode of communication between the bacteria and this larva? For that, we started with a Pseudomonas aeruginosa strain, which like it's a modified strain which lacks the one gene, and that is responsible for production of some volatiles in Pseudomonas. And we borrowed this strain from the neighboring lab actually. And then we checked the preference to the Pseudomonas, that modified strain, to the normal wild type, and we didn't find any difference. So it suggests that volatiles may not be the factor. Maybe some communication through the medium. The compounds can diffuse within the medium through the medium, so that that reaches the price as a communication mode. But just started this, and I moved to the Harvard. So some of the ongoing works now exploring this function. So here the conclusion is if the bacteria, different bacteria having differently attracting the larva, and even the larval condition, whether they are oxenic or they are exposed to some other bacteria, based on their own condition, their preference also change. Two factors controlling this. And the third work is about another fly. This is like a fat microbiome adaptation under different conditions in the shifting diet and domestication. This fly is like the common name is Avian vampire fly. It's a very interesting fly, but very disgusting flies for me. These flies actually they are originally from Ecuador, the mainland Ecuador, um, South America, and these flies accidentally introduced to the Galapagos Islands in 1960s, and where these flies found this Darwin finch nest. They are going to this Darwin finch nest or any land bird nest and laying the eggs in the nest. In the night time, these the egg hatches and the emerging larva sensing the heat in the nest because of the nestlings. Actually this breeding time everything is very well programmed. So like they are there correctly going to the nest before the like during the active season, active nesting season of the birds, and they are only ovipositing the nest with nestlings or eggs about to hatch, the bird eggs, so that they are reaching the nestlings and they are feeding on the nestling blood and tissue, so that eventually the nestlings die in the nest and these flies emerge out of the nest. So this actually very the dangerous situation against the Darwin finches, because as we know this Darwin finches are iconic finches. It's a living evidence still for the natural selection theory, according to the Darwin's theory, natural selection theory. So there is a program with the Charles Darwin Foundation. We are studying this fly to make find some control methods, some control measures. From this, so different university groups working on different aspects. Some people working on biology, some people working on the behavior, making different behaviors, and we were studying about the microbiome of the system. And yeah, this already I talked about this. So, yeah. So this there are different approaches. We now they're under consideration. One, you can see like a trapping. It's not a control method. Still, it can reduce some load. We use the fermented papaya juice as a bait for this fly, and even people tried use of insecticides, but if we can't reach all the nest, maybe in the height, we can't spray this. And island systems are very tricky, and we can't introduce all the spaces there. So it's a very restricted place for the for this kind of external additions like adding the chemicals or adding introducing any flies. It's very difficult. So use of insecticides, people proved it is working, but we can't go with that in that island. And use of natural enemy, there is a one natural enemy there against this clonus, but that work actually is now advancing. People now studying this about this the interaction between this natural enemy and the clonus in the mainland. They eventually they will try to introduce those enemy there. And the last one and best and environmental friendly method is the sterile insect technique, and we are working for this because for rearing this fly, the only requirement for sterile insect technology is you need millions of flies. In that, like we raise the males mainly, and then by radiation you will sterilize them. Then these sterile males will be introduced to the field. These sterile males then mate with the normal females, then end up with no offsprings, so that eventually this will completely eradicate that species from the particular location. So this already successful for the medfly because it's a very bad pest for agriculture. So already successful for not only medflies, there are some screwworm flies also. There are already proven method, proven environmental friendly method. So for that, rearing this fly in the lab is the key thing. But if you look the rearing now, so far the rearing is very unsuccessful. The lab, the research station, Charles Darwin Research Station in Galapagos, they have limited number of flies. They are collecting the females from the field, like trapping the females and collecting their eggs, then rearing them in the field in the lab with some like using the chicken blood instead of the normal bird blood. But the challenge is they are developing up to the adult, but they are not mating in the lab. And they are often eclosing with the mark, like deformations in the wings and all. It's like the single word is they are unsuccessful with the real. So we were looking for the microbiome, whether because many species need some microbiome or like obligate or facultative, we wanted to check microbiome. If some microbiome really needed, we can add as a probiotic to improve the rearing in the lab. So with that aim, we worked on this project and we collected different uh, like different stages from the field, from the nest, as well as from the lab colony. Lab colony is from raised from the field collected eggs. So then we checked the microbiome of this flies, and we found that the field collected, like the in the field alone, the microbiome is changing between the stages, life stages. The one thing is the larval stage, they are feeding on the blood. They are in the one place. They are not going anywhere. They are living in the nest. But in the adult stage, they are adults. They are free to feed with the other things. So we found significant difference between the life stages in this and significant difference between the male and females. And when you look the lab reared flies, and again the pattern is same. But the one thing is the larval instar 2 and 3 have same microbiome, but in the field they have different microbiome. In the same lab, even before me, one postdoc studied the only the field collected population, and they found that some host specific factors controlling this microbiome. For example, these 14 different dogging finches having different feeding habits, even they have a different morphology and they are feeding on different, like some feeding on insects, some feeding on plants, some feeding on flowers. So based on that, that also affecting the microbiome of these flies. And so we even from this from my study also I confirmed that diet as a moderator of uh, Florence gut microbiome across the life stages, and we tested different like we used some different diet also in the lab experiment, and we tried that some had like uh, some diet had effect on the microbiome, and again field collected like here this showing the male versus female separately. Here again we found the different microbiome. And the domestication actually alters the Philonous gut microbiome because, as you see, the microbiome is totally different from the field population. So domestication having effect, even though the flies are from the field, the eggs collected from the females from the field. So it means the field microbiome is not establishing in the lab. Something is preventing that. The microbes are not able to establish in the lab. That's the key thing here. Yeah, this again the same for the third instar flies. The observation is same, but the difference is little less for in the case of the third instar. So here in the domestication, I think I'm repeating some slides, right? Yeah. So finally we summarize we looked the each we trace the species level difference between different life stages and different like two different population field and lab, and we found some the bacteria which are dominant uh, in the field collected population are not establishing in the lab. For example, it's this providential species which are present in the lab in the second instar lab period population, which is not the important one in the field, but those important in the field, like for example, this Enterococcus species, Those are all in the field, but they are not developing, establishing in the lab. So this difference, this microbiome difference, maybe that's the reason. We in the subsequent visit, actually in the last visit, we tried really this splice with the isolate. We tried to isolate some bacteria and we used as a probiotic. We found the significant effect on the hatching of the larva, and yeah, that's like still the works are going on to conclude that. So with this data I have now, I summarize that life stage and diet are significant factors governing the microbiome of this plant, and adult microbiome in the field possibly reflects the foraging behavior, because we found some like Galamella, some species. They are usually commonly found with the honeybees, and they are already proved to degrade or detoxify compounds in the nectar, in the flowers. So that bacteria were in the lab, in the field, but not in the lab, and that may be the, yeah, that also the, and that showed difference between the male and female. So it means their feeding habit may be different. Maybe only the males going feeding on the nectar and females not. So that kind of predictions we became true, but it's not confirmed yet. And habitat or diet specific microbiome acquisition. It means in the lab they are easily acquiring different microbiome instead of acquiring. So that may be the reason for their ability to establish from the mainland to this island, and specifically to the Galapagos regions. Yeah. And now at Harvard, I'm working on a relevant topic, but in the same different system. And in Hawaii, actually the study system is Hawaii Island whole. In the Hawaii, there are 600 endemic species, endemic Drosophila species. They actually evolved from single ancestor species. And in the Hawaii Islands, you can see different islands with a different age. One island developed first, then next island, then next island. So the chain of islands there. You can see the pattern of this Drosophila also from one island to next island. So, and important thing, they are feeding on different, they are developing on the different substrates also. For example, the main substrates, like some developing on the bark of some tree called Molapa, and some feeding on the sap flux coming out of the trees, and some developing on the leaf, some developing on fungal bodies even, some developing on the flowers. But these all flies from the single ancestor species. So we are working this like a very good model. Whole island is a very good model. This island plus the Drosophila is a model clay or model system to study the evolution, adaptive evolution especially. So I'm working on some key questions like regarding the microbiome. Who is with whom? Like what is there with the fungal body, and what is there with the flies developing on the fungal body, and how it is related to other different substrates. So those things. And who is doing what? It's for able to do the metatranscriptome to understand the function of these microbes in this system, and like to identify what are the factors determining those microbial community. Is there any hidden adaptive radiation? Because the Drosophila adaptive radiation is very defined, very clear now. With the one species to 600 species with a totally different feeding habit, different development substrates, and I'm looking for some adaptive radiation in the microbes. I expect some adaptive radiation which may be hidden from the common eyes. We are looking only the macro animals. And how does this microbiome impact the developmental changes? Because developmental change means these different flies have different ovary number. I should tell more story to that. Yes, actually, these different flies have different ovules, and based on different ovarial number, they produce different number of eggs. Okay, for example, these fungal breeders produce only few eggs, and this bark or this leaf breeders producing a lot of eggs. So that the reproduction is a key in the evolution, right? In the natural selection, the reproduction is very important. So the developmental difference, that is the developmental difference. December we went for the sampling, and we are rearing those flies in the lab, and we are going to do some controlled microbiome studies to test the to answer those address those questions. And the second project I'm working there is about again this is about adaptation to the or specialization to some substrate. Here the fly species is Drosophila sexualia. This fly is very special. This is very close to Melanogaster, Drosophila melanogaster, or they can live anywhere. But this species is very close to them, but they are not developing in any other place. They need this fruit called morinda. It's a not it's a nowadays it becomes a medicinal value fruit, but really it's a very noxious. We were struggling with this one while in the field or while processing those samples. Really very noxious fruit. And but these flies, this Cechalia, able to live there. They are going and living, and they are successfully developing that. But the key difference is this Seychella can produce only few eggs per day. Even their overall numbers are like only I think 23 something, but in the Melanogaster overall number is 36, and this Melanogaster produce lot of eggs. So the difference, there is a developmental difference, and as well as this adaptation to the node, this Morinda citrifolia. So for that, I'm setting up an experimental evolution plan. Actually, we are using the microbes from different species which are not exposed to the Cephalaria. Then in the lab, we are allowing to evolve in the fruit, like with the bacteria, without bacteria, and we are planning to trace the mutations in the fly as well as in the microbes when you put it in the non-food. So far I confirmed, like I standardized the concentration what, because these are toxic to the other flies. So I confirmed what concentration of noni is needed for this experiment, what kind of bacteria can be used. I isolated many bacteria. Now I'm checking the compatibility between the bacteria. So then I will compile I will make a defined microbiome, like synthetic microbiome community. Then I will track them easily using the specific probes, thickness or any other probe. So because in the experimental evolution we can prove how the evolution happening specific to the particular species. Yeah, with this I come to the end, and this I would like to thank my current PA, Professor Cassandra Extraver. Is Harvard University professor as well as my investigator, and Professor Boss Eval from Hebrew University, and Professor Edward Jovovich from Hebrew Universities, and other all the contributors uh, uh, for the works I presented today. And of course, I would like to thank Professor Gopal and um, Sivakumar and Dr. Anandu for long-standing support from, I think we started our collaboration in 2014. 14, yeah. And I would like to thank each and every faculty members in the TNAU, and especially the departments and all the students. And these are like my founders from my fellowships. And this is my current lab members. We recently we took from the.
Thank you. Thank you. wonderful lecture and journey which you have traveled from the earlier part to the advanced level. Really, we enjoyed. Already, we have open to the discussion. This writing, you have asked some of the questions. I have few questions to start with. The number one is the color-based attractions. So, how did the microbiome influence the attractive color of the?
Actually, color based is actually in 1947, one group studied used different bags with different colors. In that they found this fly has some ability to differentiate the color. They are preferring some color, but they didn't confirm why or how it is happening. But in my study, I confirmed removing bacteria is affecting that ability to differentiate color.
Was it there in dark line?
These all in the dark. Visual hearing setup.
That is the light might have influenced actually.
Yeah, maybe, but the other, but the light in the room is like a standard. The variable there, we had a variable as the microbiome is variable, but all other conditions are same in the all cases. Even I put all these different color books in the same basket, same petri dish. And with limited time, I think I put only, I collected graphic like the females about to lay eggs, like very matured and only few hours, I think 2 hours or.
Another is was there any horizontal microbiome transfer from one genus to another genus? Do you have been studying
Yeah,
the microbiome transfer from different stages? Was there horizontal microbiome transfer?
yeah, it's a nice question. It's a really, it's happening actually. It's
Yeah.
social insects. It's a very common. Social insects, they are social. So horizontal transfer is very common with that. But in the system I studied, only the vertical transfer is more common. But when this in the field, sometimes we notice single fruit, mainly in the med fly. Olive fly, actually in each fly, each fruit, only one fly ovipositing egg, then it produces, I don't know much about entomology. So something they are releasing in the fruit so that other mother will not go to that fruit. So it prevents from multiple oviposition. But in the med fly, not like that. There are many can oviposit. So when if the microbes coming from different mothers and developing in the same fruit, there is a possibility for the horizontal transfer. Because their transfer is through ingestion, so it's possible.
Another interesting information was SIT.
Yeah.
SIT, sterility.
The sterile insect technology. It's a very old technique.
One of the oldest technology, even bark, why they are practicing a lot.
Yeah.
Whether here somebody is working on that, it's one of the oldest technology.
Maybe good for the this one species is
Larger
affecting
populations are
right?
spreading endemic.
So continue to focus.
Focused.
Not
People
focused.
have tried for locust.
Also call
Another bacterial sera species is very common there, right? This affecting mango. One species, I don't know the name of that species. Bacterial sera, that's the genus is called like the Gliasora.
Common name. Fruit fly.
Fruit fly is common. Fruit fly. Actually. Docile.
So I have a couple of questions. I will leave to the other audience because we can discuss it later on.
Actually, I try to finish on time. So if you have any clarification on any slide, I can go back and we can talk. Discuss.
Yeah, if needed, we can.
Yeah, that's possible because in the we try to trace the source of microbes using this. Yeah, using this core microbiome analysis, we try to trace from where these microbes come. So if something is something is here in the mother. If something, for example, here this gluconobacter, we were looking for the gluconobacter, but not there in as in the mother as a dominant. But we find using the dedicated analysis, we found that few bacteria there, this gluconobacter. The source of this gluconobacter appearing from here is actually mother. So found. But some are not. We don't know where from where this Fraturia or solitaria came. So this may be the from the fruit, either from the surface or maybe yeah. So fruit, I think usually sterile, but once punctured, then it become non-sterile. So the environmental bacteria can yeah.
Chemistry, even chemistry, chemistry might be the reason.
Because again,
They are considered in that way. Chemistry.
Yeah.
chemistry is most
Chemistry and also the interaction.
Source of microbe can be mother one that we confirmed. One is mother.
Yeah.
Second is the
Yeah.
host the host itself can. Because we didn't study the surface or anything, but I used the surface fertility fruits for the experiment. Yeah, yeah. And another thing, yeah, another thing we did this experiment in the lab, non-common lab, rearing lab, not in the microbiology or sterile environment. So I use like usually first I wash the eggs using Alconox. It's a detergent. Alconox. Alconox, Alconox, Alconox. It's a detergent from Sigma, I think, Sigma from Sigma. So 1% Alconox washed first, then followed by 1x sodium hypochlorite, and then many washes of sterile water. Like I can tell you clearly. First, like 1 minute a wash with Alconox, 1% Alconox, then followed by one wash using the previous previous, then sodium hypochlorite treatment one for 1 minute, then 3 times or 4 times sodium this previous wash. Then always I do check the sterile condition, validate that condition by putting some eggs on the media and always along with the experiment I put those. If any bacteria coming in from that in that batch, then that from the fly.
Yeah.
From the fly, yeah, that one I use for especially for the olive flies. I didn't show the whole result. Uh, for olive fly, we wanted to remove the whole gut microbiome. We use the antibiotic. Antibiotic. The actually in that lab we had already standard protocol. We proved some combination of antibiotics, and it's already proved in the lab by they tested those antibiotics. So I used those antibiotics, and I used metagenome for checking to confirm that. Yeah, only for bacteria because it's a different one. In that we wanted to kill only bacteria, but antifungal also we can use. Yeah, that is my key question actually. My the conclusion is that in medfly there are that the bacteria really favoring the polyps for the medfly because in the different fruit they are different. It's a like it's a very dynamic relationship. If, for example, if mother increasing some five different bacteria A, B, C, D, E, F, then those out of that five, only three maybe the three bacteria can establish in particular food. But other two bacteria may be in another food. So it's like different factors like control. In the medfly same. Yeah. But in the floorless, the avian fly, it's a different. In the field different, but we found the same in the lab. So the difference may be needed actually in the field, but that was missing in the lab. That was a missing part in the lab. Yeah, it was. Yeah, that is a very interesting question. Actually, this even for us, it was surprising. We used the eggs from the females collected from the field, but still, because if the mother smearing the bacteria over the eggs, then that should be developing in the lab. Actually, we expect to have those field bacteria, but they failed because maybe that bacteria needs something special. Yeah, environmental factors, or maybe we use the chicken blood, but they are already developing the nestling, whatever, bird,
Simulate the plant environment which they live.
yeah, well, there's some one group trying a lot for making them mate in the lab by changing the location, putting some trees inside the big tent. Everything.
The feed plays a major role. Feed. The feed that you are giving, it will influence a lot.
Yeah. Yeah, actually in the lab really the blood is different. Really, it's important factor because initially we used that blood from the commercial chicken farm, but later we started using blood from the free ranging chickens. We found total very found difference. Even more flies started emerging like better, yeah. So the blood is blood. I have here my email. You can ask. You can write email if you want. I have here my email. So if you have any doubts, you can. If you're asking.
So before we honor the guest, Jose, Jose, we would like to honor our analyst who has been working with us for the past 8 years. That made us to get the award. Yesterday we received the Best VCS award for the analytical services. So it's just because of his contribution. And he has been doing a lot of wonderful works, not only maintaining the machines, but also doing lots of hardware related works. Wherever the machines are out, he is moving around, even serving the Department of Environmental Science, serving the Department of Nanotechnology, Toxicology, even at Trichy. He has been to Trichy to repair some of the machines related to the chromatography. So I just wanted to felicitate him. I think he's around here. He's gone. So நம்ம டிபார்ட்மெண்ட்ல வந்து we have been enjoying a lot, availing the facilities, analytical services for free. But when you move out of the department, it's very terrific. You have to pay a lot. But we are not reciprocating what we are getting from the analytical services. We started this service in the year 2017. So since then, he is the only one serving for us, for all the students who are here, for all the faculties, for all the research fellows for free. Whatever the revenue that we have received is from outside the department. So you have to realize what the service that we are getting. Sanjeev, please. So அவரை கானர் பண்றதுக்கு we request யாராவது யங்ஸ்டர்ஸ் வாங்க. சஞ்சய் வாங்க ப்ளீஸ். Student, because you are the beneficiary, please. You can also join in. You want to speak something? Okay, see. Both
friends of other departments also prefer our department, microbiology department, for DCMS, HPLC, LCMS for further analytical facilities, even though these same analytical facilities are available in other departments, because we analyze our analytical facility will analyze quickly as possible and provide good results for other department students, for ourselves also. This is mainly presented by Mr. B. Sanjeev. our department is very thankful and very gratitude to analyze the samples and please this make service as long as possible for our for our university and our department's sake. Thank you.
You are the beneficiary.
Microbiome is more important than there of debates and written in facts. First, I would like to, it is such an honor to deliver the vote of thanks. On behalf of our organization, I first thank Dr. Arun Jose for delivering this wonderful lecture on interaction between insects and microbes. I thank him for delivering this valuable time for this lecture. In spite of his busy schedule, and I would like to thank our University Dean SPGS and Director of NRM and especially our head Department of Microbiology. And I would like to thank all for your presence and make this event more successful. Thank you all.
Thank you.